Hi there, welcome to Spec Point 2.12, which is all about mutations of DNA. So the first thing we need to know is what is a mutation. So a mutation is when the base sequence on DNA is changed. Okay, so we know from our work on the structure of DNA that DNA is made of nucleotides. As a quick recap, we know that a nucleotide is made of a phosphate, a sugar, and a base. And then we know that these join together here to another one, phosphate, sugar, base. So we've got two nucleotides now, and we know that this bond is a phosphodiester bond. Okay, we then learnt that we get double-stranded DNA because we get another nucleotide, this time pointing in the opposite direction on this side here. Okay, so then we'd have another base, sugar, this time pointing the other way, phosphate, joined with a phosphodiester bond. Now, the bases are joined in the middle here, C and G with 3 hydrogen bonds, A and T with 2 hydrogen bonds. Oops. So these are our bases in the middle. Now, if one of these bases changes, so for example, if this C mutated to become an A, for example, that changed so that that was an A, we've mutated the DNA. That's what it means. So the base sequence on the DNA has changed. Now, when we write the base sequence down, we only write the base sequence from one strand. Obviously, there's 2 strands, but we just write it from one strand. So we might say the base sequence was CATACG, and we've changed this C at the start here. So now it's AATACG. So we've changed the base sequence in some way. In this example here, we changed the C for an A. So that's what a mutation is. A mutation is a change to the base sequence of the DNA. Okay. So there's different ways that it can be changed. So we've looked there at something called a substitution, but there are actually different types of mutations. So let's go through each of those. So first of all, we've got a substitution, which just means that we swap one base for another. We've got a deletion, which obviously means we delete one of the bases, or more than one sometimes. We've got an addition, which means we add one or more bases. We've got a duplication, which means we repeat the same base lots of times, or maybe a little sequence of bases. That sequence keeps getting repeated. Or we can have an inversion, where a group of bases gets inverted, like reversed, if you like. So let's have a look at each of these in turn. So substitution is the most basic one, and I think when we first kind of learn about mutations, that's what we imagine all mutations are going to be. So as we looked for, if we had a sequence, let's put our original sequence up here, and we'll use the same original for all of the substitutions. So if this is my original sequence, remember we read the sequence in threes. So we've got ATA as our first 3, TCG as our second 3, and GCA as our third 3. And remember that each 3 bases will code for the insertion of amino acid. So this will code for one amino acid, this will code for the second amino acid, this will code for the third amino acid. So if we've got a substitution of that, we're going to swap one of the bases. So let's say we're going to swap that T for a C there. Okay, so when we come to read that, we're going to still read ATA first of all. So the first amino acid is not going to change. But then this middle section, we've changed it, haven't we? So here's our mutation. Let's underline that. So we've got a difference between there and there. One base has been substituted for another, and we're going to change one amino acid in the amino acid sequence that we make. It's not too bad. That's not too big a change. Let's have a look at the deletion now. So this time, I'm going to delete this T. So this A is now going to be here. This C is now going to be here. This C is going to be here. This G is here. This G is here. This C is here. This A is here. Depending on the length of the gene, there might be another one. So now what's happened, let's move these lines all the way down, is that rather than just changing one amino acid, which we did with the substitution, we've changed this amino acid, obviously, because we've got rid of the T, but we've actually also changed this amino acid and also changed this amino acid. We've actually changed all amino acids that come after the mutation. Now, the mutation might not occur on the very first codon, okay, it might occur later, but it's going to affect all codons, all amino acids that are coded for downstream of wherever that mutation happened. So this is where the mutation happened. We're missing the T in there. And so all the codons from that point on are going to be changed, and therefore all the amino acids that it codes for from that point on are going to be changed. And we call that a frame shift. Okay, so the ones that I put this little blue bubble round are frame shift ones, and they have the biggest impact of all types of mutation, because rather than just changing one amino acid, they change all amino acids from that point on. So addition has the same effect. So let's say now I'm going to add an extra one in. So I've added an extra A in here. So it should go ATA, but actually I've got A. Oh, sorry. That's not right, is it? So if I'm going to add one in, yeah, I could add the extra A there. And then we would carry on with the T, A, T, C, G, G, C. Okay, so I've added this one here. All right, I've added this A here, and you can see that this T has moved one down, this A moved one down, T's moved one down, C's moved one. They've all moved along one. So comparing to our top line here, remember, this is what we're comparing to. This should be ATA, but it's AAT, so it's going to code for a different amino acid. This should be TCG, but it's ATC. It's going to code for a different amino acid. GCA, GGC. It's different. It's going to be a frame shift. So it's going to affect every amino acid that's coded for downstream of where the mutation is. So it's got a big impact. The next one is a duplication. So a duplication can have variable impact. So first of all, we could have a duplication of one letter. So it might start off as it was, ATA, and then we might get duplication. So we might get 4 T's. And then it carries on, CG, GCA, etc. Okay, so we've got a repetition there. It might be sometimes that you get a little sequence that's repeated. So TA, and then it repeats TA, 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 and then it carries on with the sequence. Now you can see that the impact of a duplication depends on whether the duplication is a set of 3. So if it was 3 bases that end up being added in by duplication, then it's going to stay in sync and it's only going to affect the few amino acids. However, if it's a duplication where it ends up putting the rest out of sync of their triplet reading code, then it's going to have a bigger impact. So the impact of duplication depends on how many extra bases get added in. The last one is an inversion. So a set of bases are going to get inverted. So let's say we've got ATA as we had at the start, but instead of TCG, this time we might have GCT. That's going to code for a different amino acid, GCA. So this one, I just inverted 3 bases that were in one codon, so it's only going to affect one amino acid. But had it been a 6 that was reversed, it would affect 2 amino acids. If it was a 20 bases that were inverted, it would affect a few amino acids. But it's not going to affect every single amino acid coded up for by that from that point on. But the scale of the impact is going to be affected by how many bases are changed. Okay. Now let's talk a little bit about these frame shifts again. So frame shifts do have the largest impact. So if a mutation causes a frame shift, it causes the largest impact. So let's see what a frame shift is, first of all. So a frame shift is when a mutation occurs that alters the triplets that are read from that point on. So it's when a mutation occurs that changes the triplets from that point on. Now, the impact of that is that it therefore changes the amino acids coded for from that point on. Sometimes that's a huge impact. So it has a huge impact when the mutation is near the start of the gene. However, it's going to have a smaller impact if the mutation is near the end of the gene, isn't it? Okay, because obviously, if we had our DNA, let's imagine this is our DNA here. If we had a mutation here, it's going to change the amino acids coded for from there all the way to the end. However, if we had a frameshift mutation happen here, the impact is that it's only going to change from there to the end. Okay, so the impact of frameshift is really determined by whereabouts it occurs. The next thing is, why does it matter if we have a mutation? Why does it matter if we've got different amino acids? So let's follow that pathway through. So if you've got a mutation that changes the base sequence in the DNA, the impact of that is that it's going to change one or more amino acids that it codes for. Changes one or more amino acids that it codes for. Why does that matter? Remember that there are 20 different amino acids, and each one has a different R group, and each R group has different properties, and therefore can form different bonds. So when we then fold the protein, it will fold in a different way. So let's imagine thinking about the 3D structure, folding tertiary structure of our protein. Imagine we got this as our polypeptide chain folded in a very specific way. Remember these properties. The ones near the edge are R groups that are hydrophilic, that want to be near the edge. Any R groups that are hydrophobic will move to the middle because they want to be away from water. Any R groups that have a slight positive and then another one that has a slight negative charge will bond, pull together, because they will form hydrogen bonds. R groups that are positively charged and R groups that are negatively charged will pull together and bond to form ionic bonds. R groups that have sulfur in them, colour haven't I used? R groups that have sulfur in them, which is the amino acid cysteine, will bond together because they will form a disulfide bond. So all of these different types of bonds and hydrophilic, hydrophobic, etc. are formed because of the R groups. And if you change one R group or a few R groups, you change the amino acids, the 3D shape of this protein will be different, because if we change one of these cysteines to something else, it won't have a sulfur in. So this bit and this bit won't be pulled together anymore. So it will change the shape of the protein. And we know that 3D shape of protein is how proteins work. Their shape is so important. So the protein will form a different shape and may not work. So, for example, if it's in the active site of a enzyme, it might not be able to bind the substrate anymore. It might not work. Okay, so it's really, really important. There's one more thing to consider, which is the degenerate nature of the genetic code. So this picture here is our genetic code. Now, usually it uses the RNA bases, so U instead of T, because we actually, when we're making the protein, we're using the mRNA as our instruction, so that's why it's using the U. But remember that there's only 20 amino acids, but there's 64 different codons. So the genetic code is degenerate. So sometimes a mutation can happen and it doesn't affect the amino acid. So for example, down here, we've got 4 different triplets that code for the same amino acid. So if, for example, this U was mutated into a G or into a C or an A or a G, yeah, whatever that mutated into, that third one there, it wouldn't change the amino acid. So sometimes a mutation can have no effect at all. Let's just make sure you know how to read this kind of table. So if they gave you a codon of, for example, CCG, how would you read it? So C is the first letter, and it's C, so it's this row here. Second letter is C, so we go to second letter is also C, so it's this column here. So we're now looking at this box here. And then third letter is a G, so third letter here is G. So we're looking at this one here, so it codes for proline. Okay, let's just have a go at one more just to make sure you can definitely follow that. So let's say we're looking for UGA. So U, first letter, this row. G, second letter, so this box. A, here. So that codes for the stop codon. That would be the last one in our polypeptide. Okay. Now sometimes you might have seen it in a circle like this. It's exactly the same information, just in a different format. So let's just make sure we can use this. This is a DNA one because look, it's got the T. So let's say we're trying to find CGC. So C, start from the middle here. So it's in this quarter. G, so it's here, so it's in this section here. C, here, so it's would code for the amino acid arginine. Let's do another one. T, G, G. So T is here, G is here, second G is here. So it codes for tryptophan. So they might give you one of these genetic codes to read. And just be aware that sometimes we can have a mutation and it makes no difference at all. It still codes for the same amino acid because the genetic code is degenerate. Okay. Now, the second part to this spec point is linking it to cystic fibrosis. So we know that the context of our exam is likely to be cystic fibrosis based. So cystic fibrosis mutations, basically, there's not much to learn, except that cystic fibrosis is definitely caused by mutations. However, there are over 1,000 mutations, different mutations, that lead to cystic fibrosis. Okay, so it's not just a case of learning that, oh yeah, this G changes to this C or something like that. Okay, there are 1,000 different mutations that all lead to cystic fibrosis. The thing that they have in common, however, is that all of them affect the CFTR protein. Okay, some of them mean that it's not made, some of them mean that it doesn't work properly. Right? So in some way, it does affect this protein, CFTR protein, and either it means that it's not made or it doesn't work properly. And as we know, uh, this affects the mucus in your body. So CFTR stands for cystic fibrosis transmembrane reductants. I don't know why there's not a C there. regulator. Okay, CFTR team. Okay, so it's the CFTR gene that codes for the CFTR protein. Okay, and the impact of that is that the mucus is dry and sticky. And that obviously affects lots of parts of the body, like your breathing, your digestive system, your reproductive system, etc., which comes up in a different part of the course. But the link here is that cystic fibrosis is caused by mutations. However, there's over 1,000 different mutations that lead to cystic fibrosis. It's not just one, and that's why it's so hard to check for it. It's so hard to do genetic testing to find cystic fibrosis, because we're looking for all these different mutations. They can look for the most common ones, however. The thing they all have in common is all mutations affect the CFTR protein. And sometimes it's not made, sometimes it doesn't work properly. And then just knowing what what the CFTR stands for. Sorry, let's S off there. Cystic fibrosis transmembrane conductance regulator protein, and it means the mucus is too dry and sticky. And that's it.